stem cells used to create synthetic human embryos, a first that eliminates the need for eggs or sperm, have been developed by scientists in a groundbreaking development. These model embryos, which resemble the earliest stages of human development, could shed light on the effects of genetic disorders and the biological factors that lead to recurrent miscarriages, according to researchers. However, the work also raises serious ethical and legal concerns due to the fact that the organisms grown in the lab are not covered by current legislation in the UK or most other nations. The structures contain cells that would typically go on to form the placenta, yolk sac, and the embryo itself, but they do not have a beating heart or the beginnings of a brain. The work was presented on Wednesday during a plenary address at the International Society for Stem Cell Research's annual meeting in Boston by Professor Magdalena Ernica Getz of the University of Cambridge and the California Institute of Technology. She stated to the meeting, "We can create human embryo-like models by reprogramming embryonic stem cells. The clinical use of the synthetic embryos is unlikely in the near future. It would be against the law to implant them into a woman's womb, and it is not yet known if these structures have the capacity to continue developing beyond the initial stages." The purpose of the work is to help scientists comprehend the so-called black box period of development, which is based on the fact that scientists are only permitted to cultivate embryos in the lab for a maximum of 14 days. By examining pregnancy scans and donated embryos, they then determine the progression of the embryo's development much further along. Robin Lovell-Badge, the head of Indifferentiated Cell Science and Formative Hereditary Qualities at the Francis Crick Foundation in London, said, "The idea is that you can learn a lot about how we begin development and what can go wrong without having to use early embryos for research if you actually use stem cells to model normal human embryonic development." A rival group at the Weizmann Institute in Israel and Ernica Goetz's team previously demonstrated that stem cells taken from mice could be encouraged to self-assemble into early embryo-like structures with an intestinal tract, the beginnings of a brain, and a heart beating. Since then, a race to turn this work into human models has begun, and several teams have been successful in replicating the very first stages of development. The most recent work from the Cambridge Caltech lab has not yet been published in full in a journal paper. However, in her presentation at the conference, Ernica Getz talked about cultivating the embryos to a stage that was just above what a natural embryo would go through in 14 days. The model structures, which were each made from a single embryonic stem cell, reached the beginning of gastrulation, a developmental stage in which the embryo changes from being a continuous sheet of cells to forming distinct cell lines and establishing the body's basic axes. Although the model demonstrated the presence of primordial cells, which are the precursor cells of egg and sperm, the embryo does not yet have a beating heart, gut, or the beginnings of a brain at this stage. Before the talk, Ernica Getz told The Guardian, "Our human model is the first three-lineage human embryo model that specifies amnion and germ cells, precursor cells of egg and sperm. It was made entirely from embryonic stem cells, and it is beautiful." The development demonstrates how rapidly this field's science has outpaced the law, and researchers in the United Kingdom and elsewhere are already working on voluntary guidelines to regulate synthetic embryo research. Lovell-Badge stated, "In a way, these models should be treated the same if the whole intention is that they are very similar to normal embryos. They are not currently in legislation. This is causing people concern. There is likewise a huge unanswered inquiry on whether these designs, in principle, can possibly develop into a living animal." It was reported that the artificial embryos made from mouse cells looked almost exactly like the real thing. However, they did not grow into live animals after being implanted into the wombs of female mice. In April, Chinese researchers implanted fake embryos made from monkey cells into the wombs of adult monkeys. Some of the embryos showed the first signs of pregnancy, but none continued to grow beyond a few days. According to scientists, it is unclear whether the barrier to more advanced development is merely technical or has a deeper biological root cause. That is extremely challenging to answer. Lovell-Badge stated, "It's going to be hard to tell whether there is an intrinsic problem with them or if it's just a technical issue." He stated that the pressing requirement for stronger legislation was this unknown potential.